Meiosis and crossing over. So most of the cells in your body are somatic cells. And they are diploid or diploid, which means that they have two sets of homologous chromosomes. So humans have 23 pairs of chromosomes, and that's right, chromosomes come in pairs. And in total, you have 46, twice 23. Okay. So a pair of homologous chromosomes are called autosomes. Sometimes you'll see that word. Carry genes for the same characteristic in the same place or locus. So let's look over here at our pair of homologous duplicated chromosomes. So you have a locus right here, and let's say that the gene for eye color is at that position. If it's at that position on this chromosome, it's also at that position over here on this other chromosome. One of these chromosomes came from mom, and the other chromosome came from your dad. And the sister chromatids, this and this, are going to be identical. So sister chromatids are identical copies, but one pair of the chromosomes came from mom, and the other one came from dad. And so those are not the same. Back to our eye color thing, say mom has blue eyes. Her chromosomes are going to code for blue eyes at this locus. And dad, say he's got brown eyes. He's going to code for brown at that locus. Now that's overly simplified. Things like eye color, height, intelligence, they're all coded at many, many locations. Okay, so gametes are egg and sperm cells. And egg and sperm cells are haploid. That means they have exactly half the number of chromosomes that your somatic cells have. So each one is going to have a single set of chromosomes. So sperm, and the sperm has 23 chromosomes. A zygote is created that has the right number. It has 46 chromosomes again. So gametes are produced by a special process called meiosis, and it's going to make haploid cells. So this is an overview of meiosis. So we start here with a diploid cell, diploid, I don't know if it's a di, but diploid cell, and a pair of homologous chromosomes. So this is a matched pair. One of these chromosomes came from dad and one came from mom. So the first thing that happens is in the S phase from the cell cycle, the chromosomes replicate or duplicate. So now we have sister chromatids. They're identical. So the red strand on one side is identical to the red strand on the other. The blue strand is identical to the blue strand. Note that the red chromosome came from one parent and the blue from another, so they are not identical. The first step of meiosis will separate these two chromosomes. So in meiosis 1, the homolog chromosomes will separate. So these are haploid cells. In meiosis II, the sister chromatids will separate. This process results in 4 haploid cells. So let's look at the details of this process. So in meiosis I, we'll expect the homolog chromosomes to separate. So the core steps of meiosis are going to look very similar to that of mitosis that we studied in the section before. The differences are in this first meiosis, in the meiosis I. So in prophase, the homologs are going to line up next to each other and crossing over will occur. So in this picture of the chromosome, we have, looks like this cell has 6 chromosomes altogether, 3 pairs. And so the big one lines up next to the big one, and they will cross over and exchange bits of DNA. In metaphase II, the spindle forms. Here's the kinetochores, and the spindle forms. And we see the chromosomes line up on the metaphase plate. In anaphase, the homologs move apart toward the poles. So this is the part that makes it separate. When we did anaphase in mitosis, we were separating sister chromatids. But here in anaphase I, the sister chromatids stay attached, and the chromosomes are separated. And then we have telekinesis and cytokinesis, or telophase and cytokinesis, sorry. Okay. Meiosis II, we enter meiosis II, we have 2 cells. They are not identical. Because in mitosis, the idea was to try and make them identical. But what we did was not to make them identical. In fact, we want variation. We want them to have different genes in them. Then, in metaphase II, they line up on the plate. In anaphase, the sister chromatids separate. And we get telophase and cytokinesis. And we end up with 4 non-identical cells. And they're haploid. We do this so that when we take 2 haploid cells together, an egg and a sperm, we get a diploid organism, which is our goal after all. So let's look at this comparison between meiosis and mitosis. Start with the parent cell that has 4 chromosomes. It's got 2 pairs. In mitosis, during prophase, get my pen to work. During prophase, the duplicated chromosomes remain separate. In metaphase, they line up on the plate, and in anaphase and telophase, the sister chromatids separate. And we end up with 2 diploid cells that, if everything went well, are identical to each other and identical to the parent cell that produced it. In meiosis, we have the homologs will come together in pairs. There's a site of crossing over between them. And at metaphase, the pairs of homologs line up and one homolog goes to one cell and one homolog goes to the other cell. This makes haploid cells. That's why the 2N, they're haploid. They only have 2 chromosomes each. These 2 cells divide again, and we end up with 4 cells, and they're haploid. So both mitosis and meiosis begin with a diploid parent cell that has the chromosomes duplicated during the previous anaphase. Remind you that diploid cells have a pair of each kind of chromosome. Mitosis is going to produce 2 genetically identical cells. And meiosis is going to produce 4 genetically unique cells. And they're going to be haploid versus the diploid. Now, independent assortment of chromosomes in meiosis and random fertilization can lead to varied offspring. So you may have a sibling who has the same mother and the same father as you, and you may look similar, you may have similar characteristics, but you're not going to be identical unless you're an identical twin, and that's a whole different issue. We can arrange, because these chromosomes assort independently, we can get all sorts of different combinations. Say we have a creature that only has 4 chromosomes, so it's got 2 pairs. That gives us 4 different possibilities and combinations. Possibility A is that let's say that the blue chromosomes are from Dad. The blue chromosomes stay together and the red chromosomes stay together, and we make 2 different combinations. We have Dad's combination and Mom's combination. Possibility B is that they assort so that the long blue one goes with the short red one and the long red one goes with the short blue one. When they separate, we end up with combination number 3, where we have the long blue one and the short red one, or the long red one and the short blue one. So there are 4 different combinations. Well, we don't have just 2 pairs. We've got 23 pairs. And so that gives us over 8 million different combinations. So that's why you may look similar to your brother or your sister, but you can be quite different. So if that wasn't enough different combinations, crossing over introduces even more different combinations. So here we have an individual who has these 2 chromosomes. It has a chromosome that codes for brown coat color and black eyes, as you see here and here. On a homolog, it has a chromosome where it has white coat color and pink eyes. Note these are on the same loci. Okay, so this is a homologous pair of duplicated chromosomes. When the cells of this organism undergo meiosis, it produces, as you might expect, the brown coat color and the black eyes are always linked. So black coat, brown eyes go together, right? Well, not always, because when they're next to each other during prophase, these 2 chromosomes can exchange DNA. And if they exchange DNA, then on the new chromosome that was made, we could have brown coat color and pink eyes. So these can be separated. Crossing over occurs during prophase when the chromosomes, the homologs, are right next to each other. They get really, really close. And next I'm going to show you a picture of what that looks like. It makes kind of an X. And so it makes the possibility that you can separate two genes. That brown coat color doesn't always have to go with black eyes. And this is what it looks like when you get crossing over. Now, above here, we have a scanning electron micrograph of a DNA molecule that's in the process of exchanging DNA with its homolog.